I'd like to thank Max and Ivan for the opportunity to share our work. And I'm going to start off by telling you what you already know. But I'm going to tell it to you from the perspective of an experimentalist and an engineer. So I'm not a theoretician. I'm not coming from coding, information, or communication theory perspective. So we all know Moore's law, where information processing transformed our lives. And if we look historically, we can see that the personal computer was really a landmark case in the sense that it was the idea that they're going to take computing power out of the hands of just the few and put it into the many. And if we look at Wikipedia as ground truth, we see that that comes around 1960 when this was being developed. And since that time, we see that devices are smaller, cheaper, faster, and easier to use. And I'd be willing to bet that every single person in this room has a device on them at this point, and maybe a cell phone, and maybe even more than one. And what we're thinking about is problems involving chemical information. And chemical information is the kind of information that's ubiquitous in our natural and biological world. And if we look for a landmark in chemical information processing, I think we can look at the glucose biosensor. And this also started to be developed in about the same time as the personal computer, 1960. And the glucose biosensor transformed the way people could manage diabetes by allowing the end user, the patient, to actually test their blood glucose at home. Okay, but the trajectories between information processing and chemical information processing are very different. Since the glucose sensor, what other sensors has there been? And I would argue that there's not really very many, and most people in this audience, I would bet, do not have any device that can access chemical information. And if they do have a device, it's probably a glucometer to measure blood glucose. And so this gets us to thinking, is what is chemical information? And I think if you ask a chemist what is the chemical information in a blood sample, I think what they would probably say is that the chemical information in a blood sample is in the composition and the concentration of all the components, the chemical species that are in that blood sample. And the problem with that definition is that that says how you have to measure the information, and a lot of the analytical tools that are required to measure composition and concentration are rather complex, and it's hard to imagine that they can be miniaturized to fit into some sort of a personal device or a Fitbit type sensor. And so if we go to the information technology perspective, and Max has taught me everything I know about this, we talk about uncertainty and Baterson's definition of a difference that makes a difference. Okay, so let me just belabor this point for a second and talk about the EKG. So the clinician would probably tell you that the EKG is very rich in information because that pattern of pulses or that pattern of signals tells the clinician enough information to know if the patient's healthy or needs to be rushed into surgery. A chemist, on the other hand, might be less impressed. Yes, these peaks are associated with ions going across membranes, but it has no qualitative information about what ions, or no quantitative information about the extent or the levels, the concentrations of these ions. And in contrast, we have the glucometer. The analytical chemists look at this as a really great example because they have the ability to take all of the hundreds or thousands of components in blood, and they can filter out all the information except the one associated with glucose to give you a measurement with very high sensitivity and selectivity that gives actionable information. But from an information processing standpoint, there's a lot of information that you just threw away in this single measurement. Okay? So this is a little bit of my perspective on information. What we're really trying to do is focus on communication between biology and electronics. So it's really the transfer of information. And if we think about biology as an information processor, we know that biology can receive, can process, and can respond to information. And if we think about them trying to communicate with another cell, we show this illustration here. And the question is, how will one cell communicate to another? What modality will they use? Okay, so that's what I want to focus on now, is the modalities that one cell might talk to another. And if we think about the communication going on within your body, I'll just focus on 3 modalities. Well, we all know that the nervous system uses action potentials, this ionic electrical modality. It's integral to the nervous system. It's integral to the neuromuscular communication. But there's other modalities. And one involves very specific molecules. So for instance, hormones might be going through your bloodstream from one location where they were synthesized to some other location, to some other cell, that will have a receptor to receive that information that's stored in the molecular structure and to perform some intracellular signal processing to generate a response. And this is the kind of signal that might be involved in the endocrine system. And another signaling modality that's becoming recognized is redox. And this redox refers to reduction in oxidation, and it involves reactive oxidants that are diffusible and have the ability to move around, and it's integral to the immune system. And so let me diverge for just a couple seconds and give you, since some people may not have heard about redox and oxidation reduction reactions for information processing, let me just explain a couple examples. It's integral, this redox modality is integral in the immune system. And so, for instance, if you cut yourself, your immune cells will immediately respond by generating reactive oxygen species so that it can actually kill the bacteria that might be present in that wounded site. And so the idea is that's going to clean up the wound. But these reactive oxygen species do more than kill the potential pathogens. They also, a little bit further away from the wound site where the reactive oxygen species are less concentrated, they may provide the molecular cues to guide wound healing cells to come and start healing the wound. And it's believed that these reactive oxygen species and this inflammatory type response may be integral in communicating across biological systems, the gut, the brain, the nervous system. Okay, so there's one example. It also turns out that the redox is integral in the microbiome. We always think of the microbiome from a human perspective as the gut, but in the plant world, the rhizosphere, where the bacteria surrounding the roots, there's a lot of interactions between the bacteria and the plant cells in the root, and that's also mediated by a redox modality. Okay, so redox. So that's a modality of communication in biology. So let's go back to the story. What we're trying to do is we're trying to find ways to communicate between biology and electronics. Bio-device communication, what we want to do is to be able to detect signals coming out of the body or to observe them, and then if we don't like what we're seeing, to actually take corrective actions and in real time put information in to correct the behavior. Okay? And so what I'm trying to convince you right now is that if you want to communicate with biology, the modality really matters. Okay? So think about this for a second. What I'm asking is, how can we build a device that has the ability to observe a complex biological system when it sees that it's performing inappropriately, to take corrective action in real time to change the behavior? And I'm sure everybody in the audience, or most people in the audience, think this is a dream. This is science fiction at this point, but it's really not. It's science fact. It's actually routine, because the clinicians use EKGs all the time to measure the complex biological system. And if they don't like where it is, they can use a defibrillator to actually change and reprogram the trajectory into one that is not pathological. And these defibrillators are simple enough, if you will, that they can be implanted, and these defibrillators are actually wall-mounted on a lot of gyms, so people with minimal technical training can completely do this communication between a device and a human. Okay, so there's communication, high-level communication between biology and electronics. Now, what if we want to use a molecularly specific modality? Now we've got some problems. Well, if we want to observe what's happening, that means we're trying to measure molecules. And specific molecules, that means you're really looking at analytical chemistry. And if you want to intervene through a molecular modality, that means you're probably administering drugs. And we all know how hard it is for drug companies to develop effective drugs. Okay, the third modality is redox. Again, this is reduction-oxidation reactions, and we said that these involve a lot of times reactive oxygen species. Those are molecules, and many people who are trying to study these redox pathways are looking at these molecular features. What we're trying to do is we're trying to use the idea that these features of redox, they also have electrical features, and these electrical features are observable. And so this is the journey that we're on right now, is that we're trying to understand, can we use the electrical features of redox to establish communication between biology and electronics? Okay, and as I said, we're experimentalists, and so the way we think about it is we think about it in terms of an analogy. And the analogy that we think about is really one that's like sonar. So in sonar, a sender is used to transmit a wave. It bounces off some physical object, and the wave is reflected back. The information in that transmitted and reflected wave has information of the presence or absence of an object. If an object is there, how big, is it moving, and things like that. We're not trying to look at physical objects, we're trying to look at chemical objects. And we're going to use electrodes really as our senders and transmitters and our receivers. And the signals that we're going to send out are really diffusible oxidants. And so these are mediators. Mediators are small molecules. They are stable in either an oxidized or reduced state. What we can do is we can use an electrode to oxidize them. Then it can diffuse out in search of whatever is in that sample that we're looking at. If it finds something, an interaction is really an electron exchange. If the electron exchange occurs, then there can be a redox cycling where the oxidized species returns to the electrode and the electrode reports its redox state. And what's really interesting about using electrodes is that you can put in very complex electrical inputs to try to tune exactly how you're probing. Okay. So I said we're experimentalists. Instead of looking at sort of the theories, what we're trying to do is we're trying to see if we can demonstrate that this concept works in actual experimental systems. Okay, and the first example I'd like to talk about is really a sensing example, and this is to detect oxidative stress. And so we all know oxidative stress is bad. It's linked to lots of diseases. If you don't know oxidative stress is bad, the counter is that you know you're supposed to have a diet rich in antioxidants. So it's the same sort of idea. But oxidative stress, it's not really clear what it is, and it's not really clear what should be measured. Okay, so oxidative stress, clinical communities believe it's a significant problem linked to a lot of different diseases. It's probably operating at a global level, global systems level, but people don't really understand really the mechanisms, so they don't know what to measure. And so here's how most people would think about discovering a biomarker. Is they would say if you have a serum sample and it's from someone who is oxidatively stressed, then the metabolites in that sample should have a higher extent of oxidation. The protein should be greater oxidized. So there should be molecular features that are signatures of this oxidative stress. And so the conventional approach would be to let's do a chemical analysis, a genomics or a metabolomics analysis. Let's get the composition and concentration, compare healthy versus sick. And we can figure out if there are some specific molecular biomarkers indicative of oxidative stress. Okay? It turns out that this is going to be difficult to measure both in the discovery phase and also in the translation phase if you want to put a device into the hands of a clinician so that they can use it in the office. Okay? One advantage of this approach is if you identify specific molecular markers of oxidative stress, now that tells you what you have, and it gives you some clues of what mechanisms might be important in the biological pathological condition. Okay, and so that's an advantage. When we think about information, one of the questions I have is that you're focusing here on granular molecular details, and you're hoping that you're going to get some definitive marker of a global state. And it's not really clear that you're going to be able to go from a bottom-up approach to get sort of a global state level measurement. Okay, so what we're looking at is we're saying, okay, if the molecular features of oxidative stress really are oxidized species, greater extent of oxidized species, then these also have redox features. And these redox features can be measured by simple electrode measurements, and obviously you expect that these electrode measurements should map onto the molecular features. Okay, so you expect that to happen. That's not what we're trying to do. We're trying to map the electrode measurements onto clinical indications of disease. Okay, so we're trying to use the electrical features, access information, and see if we can provide useful clinical information. Electrical signals are simple to measure, rapid, portable, and cheap. One disadvantage is if you get a signature electrical pattern, you don't exactly know what it is. It's a little bit like an EKG. You've got a signature, but you don't necessarily know what are the molecular details of it. When we started on this project, we had information questions. How are we going to be able to generate information-rich streams to give us global patterns? How are we going to extract the information? And how are we going to be able to map our signatures onto clinical indications? So this is sort of where we got started. And the starting point was the disease schizophrenia. Everybody knows schizophrenia is bad. It's detected by clinical interviews. There's no objective measures. There's no blood tests. Oxidative stress is linked to schizophrenia, and so this was our idea that we could work to see if we could come up with a blood test that could measure oxidative stress. Okay. So the first thing we need to do is we need to establish that we can add a mediator, a chemical input, that has the ability to detect the oxidation state of the chemicals in a serum sample, and that they might actually be useful. And so the oxidant we use, the mediator, is iridium. You can see this is a scale of reducing and oxidative. This is the redox potentials of these species. These are biological. Iridium is in the oxidative region, but it's still physiological. Okay? So we're going to add this iridium. We're going to let it into the serum sample. If someone is not oxidatively stressed, we're imagining that the iridium would be able to extract electrons. So the serum of someone who's oxidatively stressed should already be oxidized to some extent, and there should be less exchange of electrons with the iridium. Okay? So this was painful. We did 118 serum samples from the Maryland Psychiatric Research Center, and basically you can see a statistically significant difference in the results for the people diagnosed with schizophrenia and healthy controls. The people with schizophrenia have greater levels of oxidative stress. So this iridium has the ability to distinguish levels of oxidative stress, and it has some differentiating capabilities. Okay? So this is just the oxidant by itself. Now what we want to do is we want to start using the electrical signal to start to enhance the amount of information we can get. Okay, so the mediators by themselves give us useful information. Now we want to add the electrical pulse sequence in to see if we can enhance information access. And this shows the experiment. We have a solution in here. This is a cuvette. You have a diluted serum sample. You have your electrode. The electrode has tiny little holes in it so light can pass through. So we're going to get an optical measurement and an electrical measurement, and these are continuous measurements. Okay, this shows an illustration. We start off by putting the reduced form of iridium. That's going to be inert. We have, as soon as we want to start the probing, we will add an oxidative pulse. We'll generate the yellow-colored iridium, so the color changes. It will diffuse into the serum sample. The sample will give up electrons. You'll get a redox cycling that will accelerate electron transfer, so you get an increased electrical signal. The optical signal will switch between the oxidized and reduced state, so we have these measurements. And so here shows all the data, but these are gory details. So I would just say that when we did buffered solutions, we were confident that we had a method that could give us reproducible results. So then we go back to the clinicians. We say, now we have a method. Give us two samples. Give us your gold standards. A person who's sick, a person who's healthy. Okay? And now we spent 3 months, and we then just took those 2 samples and tried to fiddle around with the electrical input to see what electrical input we could impose into this sample to get the greatest discrimination between the healthy and the serum from the person diagnosed with schizophrenia. We ended up with a 3-pulse sequence, a 1-minute oxidation, a 3-minute oxidation, and then a 6-minute oxidation, and then a little less oxidation. So we got a 3-pulse sequence. This is the gory details. Basically, we get output responses, electrical, optical. We could look at these behaviors. We could come up with some metrics to characterize the signals. And so for every one of these 3-pulse sequence, we got 14 metrics. Okay, 14 metrics for this 3-pulse sequence. We now are ready. We go back to the clinicians. We say, give us a larger sample size and let us see if we can distinguish healthy controls versus schizophrenia. And so we did the 5 healthy controls, the 10 people with schizophrenia. And you can see one of these metrics from the first pulse actually gives pretty good discrimination capabilities. One of the metrics from the 3rd pulse gives us some discrimination capabilities. The biostatistician combines them and says that we have a pretty good discrimination capability for the levels of oxidative stress for the people with schizophrenia. Okay? So let's just summarize what we're doing is we're trying to look at the electrical features of the redox modality. We're trying to use electrochemistry in sort of a sonar analogy to try and extract information that will give us some insights about their physiological state, their levels of oxidative stress. Okay? And so we think we've got some pretty exciting results on that. Now what we'd like to do is we'd like to reverse the arrows. Instead of taking information from biology, we would like to put information into biology and start to not sense biology, but to actuate biology. And obviously, we're not quite ready to do that with humans, so we're going to start with bacteria. Okay, so we're going to do redox to instruct biology. So let me just give you some background of why we think this is going to work, or fundamentally how it would work. We already said that we have an electrode. We can put mediators in. They have different redox potentials. They can be oxidized and reduced at the electrode. They can diffuse. Well, biology also has redox mediators, if you will. They don't call them mediators, but they have redox couples. So if biology wants to transfer electrons at a distance in an aqueous solution, it can't send bare electrons. It has to have those electrons on a molecule. And so it has to reduce something like NAD to NADH. The NADH can diffuse around, and then it can exchange its electrons. So those are the mobile carriers, the mediators in biology. Turns out that there's several redox couples in biology, and these can, in some cases, interact with our electrodes. And this shows sort of a fundamental idea of how we're going to use electrochemistry to communicate with biology through this redox modality. But it turns out that you can also communicate at a genetic level, because there are genes that get turned on in some situations by redox inputs. And so this illustrates this idea that we're going to use mediators. They're going to diffuse. They have the ability to turn on redox responsive genes. Okay, so let me just go back to the example. Imagine yourself as a pathogenic bacteria. You just fell on a wound. Life is good. You're sitting there. You've got all the food you want. The pH is controlled. The temperature is controlled. Life is good. Until the immune system starts kicking in the reactive oxygen species that with the sole purpose of trying to make your life miserable. Fortunately, your parents equipped you with some genes that could be turned on in response to these reactive oxygen species, and that those would have the ability to protect you. And so let me just summarize very quickly. There's 2 in bacteria that are very prevalent. One is the SOXR. Basically, it's a gene that in the presence of superoxide, the transcription factor is changed, and it allows gene transcription to occur for protective enzymes to degrade this reactive oxygen species. Another one is OxyR. It recognizes a different a reactive oxygen species and turns on genes for a different set of protective enzymes. And so you know that synthetic biologists can use this as a receiver part to receive redox information, and they can clone anything they want downstream. So they're going to use this. My colleague, Dr. Bentley, is using this to be able to use redox inputs in order to turn on gene expression in his synthetic biology constructs. So this shows a schematic. The electrode sends in a redox input. This electrode, this redox cycling, the engineered cells have the ability to respond to these redox signals. The redox signals are imposed because we've imposed voltages and we've added mediators. So these are imposing oxidative stresses that then turn on gene expression from this promoter. This shows the results. If we have very reducing potentials at the electrode, we're not oxidizing the mediator, we don't have this cycle, there's no current being drawn, there's no biological response. As soon as we start getting the electrode to be oxidative, we start oxidizing the mediator, we draw a current, and we impose oxidative stresses, as shown by the biological response, the expression of the fluorescent protein. Okay, and then this shows that we can quantify things a little bit. This is the charge, this is the input at the electrode, this is the biological response. Okay, and then what he does is he takes one more step. He says, let's not just use the electrode and impose the oxidative stress to make fluorescent protein. Let's put genes there that allow it to make a signaling molecule, this bacterial quorum sensing molecule, AHL. And so this will now produce a quorum sensing molecule in response to an electrical input. And then I'll have a second construct that will respond to this quorum sensing chemical signal and then respond by upregulating gene for a fluorescent protein. So input, output in this two-hop communication. This shows the imposed electrical input. This shows the biological response from what he calls the receiver cells. And then this shows, this is a FACS result. Initially, you have two populations of bacteria. Neither of them are fluorescent. They're indistinguishable. But after you impose the electrical signal, you start seeing the emergence of the distinct two populations, one that's fluorescent and one that's not. Okay, so this is the idea of using electrical inputs through redox and coupling that with SynBio in order to use electrical inputs to start changing biological behaviors. Okay, so conclusions. We think redox is a modality for biodevice communication. It's very relevant. Redox is a relevant modality. We talked about it for redox communication. We talked about it as being integral in immunity, but it's also integral in energy harvesting and other biological functions, respiration, for instance. So it's a modality relevant to biology, but it's also a modality that has electrical features that's accessible to electrochemical measurements, simple measurements. And so what we're trying to do is develop electrochemistry as a tool for redox biology and also as a tool for information sciences. We are using this tool in a redox probing way. We're trying to access information at a global level. We're generating information-rich data streams, and that we think we have a lot of versatility and flexibility to change the inputs and maybe even change the inputs in real time so that we can enhance learning. And we've got two experimental demonstrations to date, oxidative stress and synthetic biology. And then the most important slide is to recognize that how lucky we are to be able to work with such wonderful students and colleagues, both in our university and around the world. Thank you very much. Be happy to answer any questions.
Hi, with the schizophrenia samples, were you aware of the clinical histories of the 10 individuals involved? And self-medication or medication status?
Yeah, so they were all being treated in the Maryland Psychiatric Research Center. Oh, I think you might have a follow-up. They were being treated, so the clinician... That's a harder question. So these were volunteers, and then what is a healthy control when you measure... Yeah, so that's... Okay, so let me finish the first part. The people with schizophrenia, they have all of the symptomology and all of the, a lot of the data. That's not accessible to us. They're blind to us, but we can work with them to get that information. We were unable to correlate with symptoms. That was one thing we really wanted to do, but we could distinguish the people with schizophrenia from the healthy controls.
Yeah, we saw that. I'm wondering whether you might have been measuring something else. For example, they might have been medicated with Lexapro, whatever the case.
Yeah, so we had some with clozapine, so that's another drug, and we couldn't really see a difference between. So we just lumped the schizophrenia with and without clozapine together because we couldn't see any difference between that.
If there are no more questions, then let's thank the speaker again and move to the bar.